Good morning, everyone. I hope you can all hear me. Hard to tell from one of these webcasts. My name is Dr. Martin Westgate. I am the team leader in Science and Decision Support for the Atlas of Living Australia. And welcome to our first webinar for 2024. And in fact, our first one for the ALA in the last 6 months. Somehow we missed the end of last year. We're thrilled you could make it. Before I start, I'd like to acknowledge that I'm personally calling from Ngunnawal and Ngambri country in Canberra today, and I'd like to acknowledge that these are traditional lands of those people and acknowledge their elders past and present and pay our respects. So today we are thrilled to be able to be hearing from 4 speakers, 3 different organisations, about the work that they've done as part of the Australian Biodiversity Data Mobilisation Program. Now, this is something we set up at ALA a few years ago now, and it was to address something that we felt was a systemic challenge to the sector, which is that we're a data aggregator. We just store other people's data and make it available to the broader public and to researchers. Putting data in a standard format so that it can come to somewhere like the ALA is challenging. It's a bit of a faff, honestly. And so we're finding that a lot of people wanted to share data with us, wanted to contribute to an open science sort of workflow. And yet we're finding just small barriers in the way of that. Maybe that's time, maybe that's equipment. And so we established this small grants program to try and get past those barriers. And we had a lot of interest in that process. We're about to run the 3rd round of it now, but we're actually at the end of the first 2 rounds. And so we're here today to hear from 4 speakers about their experience with our program and to say about the sorts of data they've been able to share with the ALA and therefore with the Australian public and with the world as a result of that program. So our first speaker today will be David Hocking from TMAG, Tasmanian Museum and Art Gallery. Give us a wave, David.
Hello.
Lovely, thank you. We've got Mieke Strong and Dean Beasley from the Queensland Museum. Lovely. And we've got Kit Prendergast, who's an independent researcher, but working today with the WA Museum. Give us a wave, Kit. Lovely. Thanks, everyone. So look, I won't hold us up anymore. There's time for questions at the end. You viewing at home should have a Q&A portal on the, I believe, the right-hand side of your screen. Feel free to enter some questions. We'll keep them for the end, but we will try and get through as many of them as we can, though we are usually oversubscribed for questions, so we'll do what we can. David, would you mind sharing your screen and seeing if your slides come up?
Absolutely. Let's give it a go. How does that look for everyone?
It's perfect. Thanks. Over to you, David.
Excellent. I'll just stick you guys down in the corner down there. Excellent. No, thank you for having me, everyone. So today I'm going to give an introduction to the TMAG, Tasmanian Museum and Art Gallery tissue collection that we've established on the Atlas of Living Australia using the support we got from the ALA Data Mobilisation Project. Now, you're going to... Excellent. So first, I would also like to pay my respects to the traditional owners of the land that I'm speaking from. I'm on the land of the Muanina and Mumarimana people who didn't survive British colonisation here in Tasmania. And I'd also like to acknowledge the broader Tasmanian Aboriginal community and the continuing custodians of Lutruwita, Tasmania. I'm incredibly privileged here at the museum to be one of the custodians of the state wildlife collection. And for thousands of years, the Tasmanian Aboriginal people have been looking after the land and country and the wildlife of this place. And so it's an incredibly important position to be able to help to store the knowledge that we're generating around these species so that we can help to conserve them into the future. So today I'm going to give you a brief introduction to our zoology collections, and then I'm going to run through 3 case studies for how we're using our new ALA tissue collection, starting with the project that was the focus of our data mobilisation project. I'm going to run through some of the diverse range of vertebrate fauna and types of specimens we're collecting, how we collect and store those samples, and then what sorts of new analytical approaches these specimens are being used for, which is a bit different to your traditional museum collection. And then I'm just going to touch on what's in the pipeline for where we're taking the tissue collection next. So to begin with, Tasmanian Museum and Art Gallery is the state repository for Tasmania's state wildlife collection. So we have an ongoing record of all of the species that live here in Tasmania and in some of our subantarctic territories like Macquarie Island. And this is an essential reference collection for ongoing research and conservation, but it's also a source of samples for the new types of advanced analyses, which have really come into their own in the age of genetics. So we have three main zoology collections. We have an enormous invertebrate zoology collection with over 180,000 specimens. We have a geology and paleontology collection, and I wear the hats of both the vertebrate zoology and paleontology collection, hence the dinosaur cap. And then we also have a vertebrate zoology collection with more than 20,000 specimens. But today I'm going to be talking about our frozen tissue collection, which has about 30,000 specimens and is one of our biggest, most rapidly growing collections here at the museum. So our frozen tissue collection is primarily made up of tissue samples from vertebrate animals that are stored in ultra-low temperature -80 freezers. And these are samples which are often things like a muscle or a skin sample, and they're available for destructive sampling. Oops, I'm doing anything. All right, have you guys still got me?
Yeah, we can see you.
Wasn't you guys? Excellent. Sorry, that was the webinar thing. I think it was the Teams meeting popping up telling me I wasn't in. Alrighty, sorry about that. So we have this tissue collection with frozen tissue samples that are available for a range of destructive sampling approaches, including things like genetics, but also toxicology. And I'm now going to run through 3 case studies, starting with the one that we used for our data mobilisation project. So to begin with, I'm talking about the Tasmanian devil tissue bioarchive. So Tasmanian devils are the world's largest marsupial carnivore after the extinction of the Thylacine, and they're a scavenger with these amazing teeth that are adapted for crushing bone that allows them to capture and eat all of the prey they're trying to consume, which makes them a really important and key part of the ecosystem in the Tasmanian landscape. But unfortunately, they've also suffered from a rare contagious cancer that's really ravaged the devil populations. It was first encountered and recorded in 1996, but then a second strain appeared in 2014. This is a fatal contagious cancer that's led to an up to 80% decline in the population in the areas where DFTD is present. The pictures at the bottom show you a skull from a Tasmanian devil that really shows you the effect this cancer has on the animal. So on the left, you have what looks like a healthy skull with all of the bone preserved. And on the right, you can see where the bone has been resorbed by the lesions. And in particular, this dissolves the bone around the teeth, which means the teeth end up essentially hanging in the soft tissue so that the animals aren't able to feed. It really is a devastating disease. And it's really crashed the population of one of our most important vertebrate species here in Tasmania. And because of that, the Tasmanian Devil Tissue Bioarchive was established by the Save the Tasmanian Devil program, which was managed by what is now the Department of Natural Resources. And this is a collection of specimens that were collected and stored to enable research into the genetic evolution of DFTD. So it's a long-term collection of things like ear punch biopsy samples that allow you to track the genetics of the individual animals, but also tumor biopsies that allow us to track the genetics of the tumor independent of the host itself. All of this data is recorded against a microchip number for the individuals over successive years, which means we might end up with tissue samples from the same animal in multiple years. And this is a collection that's then been accessed by researchers from all over the world to study this rare contagious cancer. On the bottom, we've got one of the traps on the far left that's used to capture the devils, and a pretty chilled looking devil sitting in the trap. We also take morphometrics, and these samples are mostly collected by the Save the Tasmanian Devil program, which is run by NRE. So their work is to take morphometrics of the animals, like tooth shape, and then we track all that data against a microchip number, which is the device you can see on the far right that's used to insert a microchip under the skin. So this collection was fairly recently transferred to TMAG, to the Tasmanian Museum and Art Gallery, to help facilitate the ongoing maintenance of that collection and also to provide access for researchers along with the rest of the state collection. And so this saw the transfer of a really large collection of frozen tissue samples, including things like ear biopsies, but also blood serum sample and histological preparations. And it included a large bulk collection with about 90 cryo boxes containing more than 8,000 sample vials, and a histology collection with about 1,600 formalin-fixed specimens. And that became the focus of our data mobilisation project. So one of the things that happened, especially for the histology specimens, they were prepared in the Tasmanian Wildlife Health Laboratory. And as part of that process, they were given a laboratory number. And through that process, some of the data that had been stored against the microchip number was then disassociated from the specimens that had their lab identification number, which meant that we had some really important specimens, but we sort of lost some of that connection. And it was a lot of work to then reconnect that data. And that's, as you were saying earlier, that's a pretty big job to actually go through those spreadsheets, work out what belongs where. And so that was sort of a core part of what we did with our data mobilisation support. So the metadata for all the records was then curated against the Darwin Core standard. And then we were able to mobilise not just that sample, but our entire Devil tissue bioarchive, which adds up to about 10,000 records that have gone into the TMAG tissue collection. You can see the spatial spread of those samples on the map on the left, and then an example of one of the just one of the samples. This is actually a heart muscle sample. It's stored at -80. It's from a female animal, and in our other catalogue numbers, we have both the microchip number, the LIMS number, the lab ID, and then we also have the new catalogue number for the Tasmanian Museum collection, which is the number that sample will then be referred to in all future publications using that sample. Because I'm talking to you from a museum, I couldn't go past showing you some bad taxidermy. So Tasmanian devils are remarkable animals, but in the past they were called devils because of the scary noises they made and sort of the reputation they earned. Actually, these are really timid and delightful animals, and this is a devil that was hiding under the car for about 20 minutes after it got released from one of the traps before it got brave enough to run back into the woods. So they really are an amazing species. The next case study I just want to touch on is marine megafauna strandings. So Tasmania is a hotspot for marine mammal strandings in particular, because we're at the intersection of a bunch of different currents. We have the Leeuwin current coming from Western Australia, the Eastern Australian current coming down from the tropics, and then the Southern Ocean and the circumpolar Antarctic current below us, which means that we end up with a really interesting and diverse range of species from a wide source of areas that all wash up in Tasmania, including things like baleen whales, rare deep sea beaked whales, the Antarctic seals, and also tropical sea turtles. So to give you an example, in 2022, we had a mass pilot whale stranding on Ocean Beach on the west coast, and it saw about 250 animals come ashore. Fortunately, about 50 of those animals were able to be released back into the wild and got off the beach and hopefully survived. But that left about 198 whales on the beach. So our team from the museum collected samples from those specimens for our tissue collection. So we collect a blubber and skin sample, and you can see that in that central image, that thin layer of skin and the thick layer of blubber below it. And that can be used for extracting genetic information, but we also collect a broad range of metadata, including the sex, the age class, body morphometrics, and the lactation status, which of the females are actually producing milk. Interestingly, we didn't find that many really young animals, but quite a lot of the females were lactating. So it tells us interesting things about whether or not we're missing some of those animals, those young individuals that either may not have stranded or we may not have found them. What these types of samples can allow us to do is then look for some of the patterns in the population dynamics of these animals. And this is actually the second of a really mysterious set of strandings. So in the year 2020, there was a much larger stranding with 470 animals on the exact same beach. And it was 2 years to the day that the 2022 stranding happened. And if we look at the makeup of male and female animals, we can see that actually the proportions of the structure of those groups seem to be very similar. And so by looking at things like the metadata around those animals, but also then the genetics, we can start looking for things like whether or not there are relationships between those two groups that might help to provide some clues, because it really is a mystery why these animals are stranding on these beaches, and to have the two strandings at the same spot exactly 2 years apart is really bizarre. Some of our marine mammal specimens are also about to go into a new project being run by CSIRO for the National Biodiversity DNA Library. So this is a new project where they're building a reference library of sequences from marine predators for future environmental DNA analyses. And so for that project, they require both a tissue specimen, but also a physical voucher specimen that's identifiable. So something like a scan or a study skin for a seabird that's held in a museum collection that gives you a way to verify the identity of the DNA that's come from that specimen. So this is a really exciting project that I know a bunch of museums in Australia are providing new specimens for. Okay, the final project I wanted to touch on is the TMAG Threatened Birds project. So the first two things we've talked about, the devils and the marine mammals, one of the main uses for the tissue samples is to look at the genetics of the animals that we're talking about. But this project has really come into its own by looking at tissues from a totally different perspective. So as part of our Threatened Birds project, we're looking at the causes of mortality in our threatened birds of prey. So in Tasmania, raptors face a really broad range of threats, including things like vehicle strike, but also collisions with power infrastructure, like wind turbines and the electricity lines, and also poisoning by things like rodenticides and environmental pollutants like heavy metals. We're collaborating on this project with TasNetworks, the power supplier in Tasmania, and also Woolnorth Renewables that manages some of the wind turbines, to help identify the causes of death, to help them with the mitigation of that infrastructure by putting things like bird flappers on power lines to help prevent more deaths. For every bird that's collected, we're undertaking a gross necropsy and recovering samples. And the image at the bottom there shows the use of a forensic torch, which fluoresces burnt feather, which allows us to identify electrocuted birds, even if the signs are really, really small. This is particularly useful for birds that land on the wire that might have burns on the pads of their feet that aren't immediately visible to the naked eye, but they fluoresce brightly under this forensic torch. So if we can confirm that electrocutions occurred, that triggers mitigation on those lines. So in addition, we're collecting frozen tissue samples, things like liver samples and muscle samples, and also bones for the metals. And this is being used for a genetic analysis of body size with collaborators from UTAS. And we're also then running analyses looking for rodenticide poisoning and also heavy metal contamination, and in particular lead. And we're producing this alongside traditional museum preparations like osteology and study skins. Based on the current trends, we're seeing about 40 wedge-tailed eagles coming into us per year, about 5 white-bellied sea eagles, and about 8 grey goshawks. So this is a really, it's quite tragic to see that many animals that are essentially coming to us through different forms of human-wildlife conflict primarily. And so by being able to work out, so for instance, the information we're getting about rodenticides is actually quite significant because we otherwise wouldn't know that some of these species were so heavily affected by these. And this is something that we could put in new regulations that might be able to help to mitigate that. This data set, our threatened raptor data, is the next data set we're actually preparing to upload to the tissue collection on the ALA. And so the samples that we've collected, we've already run the heavy metal and the rodenticide analyses for most of our raptor data, but we've still got samples that can be used for other things. So we've had a collaborator contact us to look at the fire retardant PFAS chemicals. We've also had people look at things like the intestines from our birds to look for gut parasites in Tasmanian eagles. And we have a huge collection of samples for the diet from stomach contents for these birds that we're yet to actually run. So that's another project to look at the diet of the eagles and how that may inform the route of things like heavy metals and rodenticides into these birds. All right, so a couple more things. So the next step for our tissue collection, we're going to continue routine collection of samples for all of the new wildlife we're preparing here at the museum. And we're going to continue to collaborate with researchers in particular to try to identify the needs for future research. So what types of specimens we should be collecting that might be useful in future. And then researchers often build collections through their projects. So if they want researchers in future to be able to access those, it's a good idea to get in touch with your local natural history museum, and it might be possible to lodge those specimens so that they then become part of the data that can be reanalyzed in future and potentially for different purposes. So to contact us, if you want to use any of the samples, if you want to talk to us about getting access to Tasmanian vertebrate material, and also keep an eye on the ALA tissue collection, which we're continuing to migrate new data onto. And then the last thing I want to touch on is some of the ongoing challenges that we're facing with this project. I guess this is probably a problem for everyone, but we're going to have a continuous issue with freezer space as we continue to expand and grow this collection, and then finding funding for new facilities is really important. But one of the big things that these projects require is the staff time to carefully curate and prepare the data, in particular so that we can migrate stuff onto the Atlas platform. And so that was one of the reasons why the ALA data mobilisation project was such a helpful project for us, because it allowed us to put the staff time in to be able to get the devil tissue bioarchive as the first instance of data across onto that platform. And we're looking forward to potentially applying in future as we continue to expand this collection. That's everything I had for you. And this is an echidna that visited my house the other day. So I thought I'd leave it there.
Thanks, David. That's fantastic. And that's a beautiful echidna. Great work. Our next speakers are Mieke and Dean. Feel free to put your slides up, David, if you could stop sharing. We'll keep questions for the end. Like to anyone watching along at home, feel free to add questions in the Q&A. Really interesting talk there, and a fascinating opportunity for some events that are actually slightly tragic in some ways, to inform future science despite their tragedy. I think that's really interesting. Now, who's running slides next? Dean. Lovely. Okay, over to you both. Looking forward to hearing about it. Thank you.
Thanks, Martin. I'm Mieke Strong here, and would also like to thank David for kind of creating a really great segue into our talk by mentioning the parasites of the birds that you've got in the TMAG collection. So for the project that Dean and I were involved in at the Queensland Museum, we were mobilising the Thomas H. Cribb Parasitology Research Collection. So you may be wondering what our connection is to this, if you wouldn't mind, Dean. So I did my PhD on marine parasites on the Great Barrier Reef under the associate supervision of Dr. Tom Cribb. And it was not everyone's cup of tea to do parasite research, but it definitely was ours, and especially in locations like these. So let me tell you a bit more about the collection itself. So the Thomas H. Cribb, which we are going to refer to as the THC, Parasitology Research Collection, it spans over 40 years of collecting data from Tom himself and all his collaborators and students that have come along the way. And it represents 8,500 type and voucher specimens. So this is like representations of species names, the individual samples that are stored in the Queensland Museum and also other institutions around the world. It represents 20,000 fish host examinations with 18,000 trematode parasite species. So these are flatworms samples from those fish. And that covers 2,760 host parasite location combinations. It's a huge, massive collection. And all the data at the time when Tom was retiring at the end of 2021 was in Microsoft Excel spreadsheets and hard copy dissection records. So because Tom was retiring, moving his collection from the University of Queensland over to the Queensland Museum, this funding opportunity was perfect to actually get this data shared with everyone. So next slide, please. What we did in this particular mobilisation project was we consolidated the data that we had with the Excel spreadsheet and a lot of the data that was not collected into the spreadsheet from the dissection records. We cleaned it up for consistency. And for those non-taxonomists out there, we also checked that the names that were recorded over time were then aligned with the current taxonomy, which does change over time based on greater understanding of the animals that we're talking about. So we validated against the World Registry of Marine Species, that's the worms database, and kind of cool name, things like that, we're looking at worms, and also the Australian Faunal Directory, which is the AFD. We checked that the records that we were going to supply to the museum were not the same as ones that we already had at the museum, because we don't want to double up on that information. And then we looked at all the data that we had from the THC collection and then map it to the Queensland Museum. That's QM Vernon. That's a software system, collection management system that they manage all their data there. So this investigation into this greater data source allowed us to expand the Queensland Museum occurrence data model, which I'm going to talk about a bit more further, and also then expand the data that the Queensland Museum as a whole then sends through to the ALA. Slide, please, Dean. So let me just use this very simplified model to explain things a bit further. What we've got is a host, so a fish with a THC identifier number. And then we've got the parasites that were extracted from the fish in this little tube here. And we've got a few different scenarios of what we're sort of distilling out from the data itself. So there's a situation where you've got a fish that has no parasites found in it. We've got a fish that has one or two or many more different types of parasites that were found in it. And then we've got a certain dissection protocol that actually combined a few fish together and found 0 to as many different types of parasites within that dissection protocol as there was. And then what we had to do with all this great diversity of data is then balance that from the raw source data into what the traditional sort of Queensland Museum data model that is shared is based very much just on the specimens that is housed within the museum. But if we did that, just like focusing on the specimens, then we'd really lose out on all that diversity of what's going on with the hosts. Like, what about those hosts that didn't have parasites? So we came up with this new model that we could get data on all these aspects. So the worms that are already in the museum, they have numbers that have got a G prefix, so that's for invertebrates. So they're sort of represented with this slide over at the far side, say G1. But we've also got all these potential specimens in the Tom Cribb collection that just need to be double checked and verified that they're worthy of going into the collection. They haven't dried out over time. If it's a 40-year-old specimen, it might not be quite as good as it could be, or there's like lots of replicates. We really want to make sure that what goes into the museum as proper verified material is, proper verified material. But we also want to collect the value of what was actually found in these fish. So I've got this numbering system called GB. So this is like a bulk collection where we can tease out those validated specimens. So these GB numbers are they are known, but they are not yet validated. And then in association with those GB numbers are the GO numbers to cover the hosts. So this is an occurrence specimen which is associated with the worm parasites themselves, or they may not. Like in that first occurrence situation where we've got a fish host that didn't have any parasites. So this new model, we can collect all that data, fish, the worms, and then the actual verified specimens that can increase over time. And we get to also combine all these relationships with this expanded model. So I'm going to throw over to Dean now, who's going to talk to us more about what we did in the actual Vernon system to expand the data model relating to Darwin Core and onto the ALA. Thanks, Dean.
Thank you, Mieke. So in Vernon, we had data in spreadsheet format essentially, and we ran some imports via XML. So we had 20,000 host to current records, and we ran imports, about 3 imports of 10,000 rows each. Sorry, 2 imports, I should say. And then in parasites, we had a bit more data, so we split that into 3 XML imports of about 6,000 rows each. And then we came back and we actually updated 17,900 records to build the relationships between those bulk records and those occurrence records within Vernon as well. In terms of changes to the CMS, the existing XML export that we were using to send data to ALA consisted of about 45 Darwin core fields, and during this project, we sort of boosted that to about 63 Darwin core fields. So we've reconfigured and added new fields in Vernon, including associated taxa, occurrences, other catalogue numbers, field numbers. We've got verbatim event dates, verbatim Latin long, and some additional taxon rank, taxon fields, so taxon rank, taxon ID, which we can draw from systems like worms or AFD, subfamily, subgenus, and like. We've also added some additional locality information, continent, country code, water body, island, and municipality where that's applicable. Vernon browser, Vernon CMS, I should say, has a feature called user symbolics. They're kind of like user created fields. So we can do calculations or joins, extractions behind the scenes to build content and send it out in different ways. And we use that extensively for our online delivery of records, be that through one of our Vernon browser portals. So we have 2 different Vernon browser sites running. One that hosts our publicly accessible collection records. So there's about 820,000 records publicly accessible via our Queensland Museum online collection. That includes cultures and histories records, biological specimens, and geological and fossil specimens as well. And then we have an entirely separate instance just for ALA data, which CSIRO comes in and harvests data from that system. Now, that was quite old, and you might see there's a screenshot there showing what our Vernon Browser 2 was. This is now the current Vernon Browser 6. This project presented an option to upgrade that infrastructure. When we went and talked to Vernon Systems, who provide our database to us, about adding these extra Darwin core fields, this Vernon Browser 2, because it's quite old technology, didn't allow for that updating or addition of extra database fields on the fly. And to make matters worse, it was kind of end of life anyway. They're not supporting anything earlier than Vernon Browser 4 now. So we made a decision, let's upgrade and put our data across into Vernon Browser 6. So this new Vernon Browser 6 was set up for QM. We rebuilt our Darwin core. Sorry, I should say we added those extra Darwin core fields and rebuilt our XML export with support for these additional fields. And the other thing we did is that we changed the field names on the Vernon Browser 2 so that they match the Darwin core fields, which would streamline the harvesting of this data. In Vernon Browser 2, CSIRO had scripts that ran directly on the actual database server to extract, package up the data and send it. This new Vernon Browser 6 has a web services layer, and harvesting occurs via an API. We've reloaded the entire data set of 900,000 records and approximately 16,000 images to Vernon Browser 6. And then in some cases, we've revisited records and reloaded them. We were sending all of our other catalogue numbers to one field, but we've decided to split that into 2 different fields, other catalogue numbers and a field notes field, after talking to CSIRO about the best way to do that. So new scripts were required to do all this heavy lifting of harvesting via the API. And now I must give a big shout out to both Rose and Mahmoud from CSIRO, who've done some wonderful work to learn and understand how this API works and get the best out of it. In the early days, the extraction was taking the best part of a day to get those 900,000 records out of the system. But I think now with some optimization, we've removed the problem of duplications, and we've got that harvesting down to about 4 hours now. So thank you so much for that excellent work to get the information out of Vernon browser. This just shows a map of what QM data looks like on ALA. And this is our mapping of our 38,000 records of the Thomas Cribb parasitology collection. So there's not dots all over the place, but the collections took place on many islands along the coast, as you can see, and they visited similar locations. So there's lots of occurrence data, lots of fish bulk trematode data from those particular locations around the coast. And that's about it. We would like to obviously acknowledge the contributors of this wonderful collection, Thomas Cribb, of course, Terry Miller, Scott Cutmore, and Nick Wee. So we've had any number of meetings, the 4 of us, trying to troubleshoot this data model and get the best out of it for getting this data to ALA. Mieke and myself, these are our email address should you have any questions and would like to know more, and there isn't an opportunity to do so in this next follow-up Q&A session. That's all from us, Martin. Thank you so much.
No worries. Thank you, Dean, and thanks, Mieke, as well. I mean, what a fantastic resource that you guys have got access to there, and the work you put into sharing that's next level. It seems like every step of that process took a lot of work. Thanks so much for that. I'll remind everyone again, feel free to load questions into the chat. We've got 10 or 15 minutes at the end of this session where we'll have plenty of time to discuss them, so feel free. Kit, over to you if you'd like to share your screen.
Yep. Okay. Good morning, everyone. And you might have seen I have an interesting background. So I live in Queensland now, but most of my research has been done in Western Australia. And I'm back here in WA at the moment, doing a bit of work, couch surfing at my friend's place, who is a taxidermist at the WA Museum, hence the taxidermy. So interesting background. But yeah, so I am so incredibly grateful for the grant that I was awarded, because it has really made me be able to put some incredible bee biodiversity data onto the map of Australia, essentially. So I am a native bee ecologist and have conducted a lot of research across Australia, but mainly in Western Australia. So yeah, I did my PhD in Western Australia, but have been since doing research in other parts of Australia, now live in Queensland. And I have done diverse things, including biodiversity surveys on traditional landowner lands, in urban areas, in Stradbroke Island, Tasmania. I know we had like our first presentation from Tasmania, so I did some research in Tasmania a few years ago. I've done a bit of research in Queensland too, in Sydney. So native bee data all across Australia in all types of habitats. As I mentioned, people's gardens, like my PhD was about biodiversity in the urbanised region of southwest Western Australia, but I've just done apple orchards, as I mentioned, like traditional landowner lands, mining rehabilitation sites, so a whole range of habitats and places across Australia, discovering the native bee biodiversity there, of which we have an incredible amount. So my collection of bees for southwest Western Australia over the years. So I started my PhD in 2016, and I was surveying 14 sites over 2 years, monthly, in spring and summer. Then I conducted research for Department of Biodiversity, Conservation and Attractions, surveying 4 sites over 3 years. I've also done surveys for local councils, friends groups, City of Bayswater, Friends of Lake Claremont, Allison Band Reserve, Walyata, which is traditional landowner land near Broome, in Pernalulu, which is the Bungle Bungle Ranges, in Mount Claremont, for Chapman River National Park, which is near Geraldton, in Carnarvon, where I was studying Amygdala dorsoni, Coolgerrinup Reserve, which is in southwest Western Australia, along Kwinana, the Row 8 Rehabilitation Corridor, Lake Johnston, where I was surveying a critically endangered plant for its pollinators, which is out in the middle of nowhere essentially, and many more places. So over all these surveys, I've collected a huge diversity of native bees, and as you can see, here's just a small collection of some of them, and as you can see, they're very, very beautiful. And during my surveys, I've also made new range records, many species that are undescribed, new host records, so a big fascinating data set. So these were thousands of specimens, and over 300 species, species that have never been recorded on the Atlas of Living Australia. So when you go to Atlas of Living Australia and you look up a species, there'll be no actual records on the map. Many undescribed species, almost every single survey there are undescribed species. So all these records I had were not on the Atlas of Living Australia. So that data, useful to me, but not useful really to anyone else. And they also weren't properly catalogued in the museum. If you get to go to the collections, entomology collections in the WA Museum, there's, I think by now, 2 cabinets that are just my bees. And as we heard actually from previous talks, a big issue is not enough space in the museum, not enough staff to curate all the specimens. So there was this amazing collection of bees that wasn't even data-based, wasn't catalogued, was just sitting there. And this is true of many museums and many collections. So I was so incredibly lucky and grateful to get this grant so that this incredible collection could start to be data-based. So doing it is very boring and also very time consuming, and you need like a lot of concentration. So you first have to pin all the specimens. Some of them are just over 2 millimeters long. So you have to use a pin and a little pinning point and glue it on and make sure that the bee, is stuck in the right way. ID all the bees, and this isn't an easy task. So most genera don't have keys. They were described maybe 100 years ago, before photography. The descriptions are very poor, and so it's very challenging. So I then have to label them with all the information, cut the labels out. Many of the labels I needed to recut so that they could fit better within the museum drawers. Then arrange all the specimens into species groups, then transfer all the data from the labels into the spreadsheets that is then transferred into Darwin core format. So there's a number of steps between writing the labels for the pinning labels, transferring into the catalogue data set spreadsheets for WA Museum, then transferring it into the Darwin core. Have to then, for all the specimens I have, add catalogue numbers to them, which are special numbers that are associated with the WA entomology collection. So for all my collections, each specimen has its own unique identifier number, but then I have to add an extra catalogue number for the WA Museum. And then for some of the specimens, I've been starting to barcode them. So again, we heard a little bit about genetics earlier on, and so most of our native bees are still, not barcoded. There's no barcode associated with them, so I've started doing that as well. So if they're barcoded, I add additional label and additional bit of data so that we can go back and find them if need be. So it's a really long, arduous process, but I'm really happy that I've been able to come back to WA, because as I mentioned, I live now in Queensland, and all my collection is in Western Australia. I've donated a few specimens to the Queensland Museum, where I've been doing some research there, but those specimens haven't been catalogued. So same scenario there, not enough funding, staff to actually database the specimens. So they're just sitting in the Queensland Museum. Hopefully one day they'll also be databased. But yeah, the bulk of my collections are all in Western Australia. So I've been able to come back to Western Australia, spend days at the museum, databasing them. And I think by now, 7,000 records are now in the WA database, but this is just the tip of the iceberg, but it is a very important tip. So some of the key specimens that I have focused on databasing because they're really important was this bee species. So it had been described as 2 different species, one from the male and one from the female. The male was, the type was in Western Australia, the type of the female was in Queensland. And thanks to doing some, the females have never been collected in Western Australia, so that's why it was always thought 2 different species. But whilst I was doing my collections, I was collecting these males and females, and I knew that they were the same genus, and because I was collecting them concurrently, I thought they were probably the same, barcoded them and found that they were the same. But then when I was checking the barcodes, I realized it matched another species. So then me and my colleague, James Story, we synonymized the 2 species. And when you're doing any sort of taxonomic paper, it's really important to have those specimens catalogued at the museum. So I was able to catalogue and put on the ALA all those species. And I don't believe that the taxonomy has been updated on the ALA yet, but they're going through a taxonomy update. And so these 2 specimens, species, will be combined as Xanthesma, Xenohesma, Brachycera. And so they're now all database, which is really, really good. As you can see, they are very fascinating, beautiful little bees. Then Leoproctus zephyr. So Australia has 1,660 described species. Leoproctus zephyr was the 1,661st, but then we synonymized those 2, so we reduced the number of species. But there are hundreds more that are undescribed. This species, I found it during my PhD surveys. It was one of many that undescribed, but this species was very distinctive because it had a protruding clypeus, which all the press latched onto and said it had a nose or a snout. Bees don't have noses. They smell with their antennae, but had a snout that's sort of like a dog snout. But I love my dog, so I named this new species after my dog, Zephyr, who is in the photos there. She's a Maremma. And this species is very special. It only occurs in southwest Western Australia, in bushland, no residential gardens, and it is very, very specialised. I've only collected on Jacksonia sericea, one instance of Jacksonia fursulata, and there's a previous record. So this has been collected, I think once or twice before, but was sitting in the museum as one of the many undescribed species. So very specialised in its host associations, only recorded in, I think by now, 7 locations, and obviously only where Jacksonia sericea is, and there's also many places where Jacksonia sericea is, but I haven't found it. So rare, specialised, was undescribed. So I felt very passionate that I needed to, describe it so people are aware of it. Hopefully people can start conserving the bushlands that it's in, planting more of the flowers. So yeah, I decided to, in my spare time, describe a new species and publish that. And again, of course, really important to have those specimens that this new species is based on catalogued and on the record. So hopefully all of those have now been databased and uploaded to ALA. But as I mentioned, I think ALA is like updating their taxonomy. So Leopropter zephyr, I don't believe is currently on there, but will be once they update the taxonomy. So that's very exciting that this species can finally be on the map and is now secure in the WA Museum. But there are many, many specimens in the WA Museum that are undescribed. You know, ones that I've collected as well as ones that other people have collected. So it is a big task databasing the species and specimens, as well as needing to describe them. So whilst I was also doing this databasing, I was databasing the Walyata specimens because I believe it's very important to have those, especially considering that they're collected on traditional landowner land, being able to have them you know, publicly available, so people, including traditional landowners, can access that data, and also specimens that were collected on the sort of near mining land of that critically endangered plant. Really important database those. So I focused on databasing those as well. And two of the megachili bees that visited this critically endangered plant were undescribed. And so I have databased them, as well as have started to do the initial steps in finally describing them. So taking high resolution photographs and measuring all their features, you know, identifying what makes them unique. So these are the two bees here. They're megachili. They both have really interesting clipeal structures, as you can see, sort of like big shelf, and then these big prongs. So I'm when I find time, I'm going to try and describe these two and add them to the, you know, the growing list of Australia's native bee biodiversity. So future challenges. So I've always just been chipping away at this. Museums are very understaffed. I keep collecting, so I keep on, you know, I still am doing surveys for various councils, and so the collection is growing, and you know, it's really a full-time job, I think, to database everything in the museum. Taxonomy of native bees is still very poor. And then there's also lots of records on ALA that are coming through iNaturalist that are actually incorrect. So to be like verified on iNaturalist, you just need 3 people to agree. But when I look on ALA and see some of the photos, they're incorrect. So taxonomy is very underappreciated, underfunded, even though it's the backbone of biodiversity. So there's a lot of challenges ahead that I'm very grateful for the ALA that I've been able to start resolving some of these. A little bit of self-promotion. I'm going to Adelaide today, and then on the 23rd and 24th and 25th, I'm doing a fringe comedy show called The Birds and the Bees, all about native bees, to try and raise awareness about pollinators and our bee biodiversity in Australia in a very alternative, fun format, because I found that there are so many people out there that don't even realize that we have native bees, or the challenges that native bees face. So I created a comedy all about bees, which is very fun. So if you know anyone in Adelaide, or if you're in Adelaide, I'd love to see you at Fringe. So thank you, and big thank you especially to the WA Museum, which is, they've been so good in letting me, you know, store all this collection there, and you know, be able to use their resources and microscopes and everything. Obviously, Atlas of Living Australia and CSIRO for supporting me and being able to make this possible, and especially a big thank you to Dr. Nick, who is the curator of entomology at WA Museum, who has been so helpful to me and let me come in, use the desk space, and have all my bees stored safely there. So really appreciate it. Thank you.
Lovely, thank you, Kit. What a fantastic talk. And I'm from Adelaide, so it's great to see the Fringe mentioned, though I'm not there now. Good on you for that. I might just start with you, Kit, if that's all right. I guess we've got about 5 minutes for some questions now before the system kicks us off on the hour. So you mentioned that a lot of this was from your PhD, and my understanding of entomology is that this is a systemic problem, right? Where it's very normal to collect hundreds or thousands of specimens during an entomology PhD. Do you think there are changes that we need to make to research in Australia to make this work easier? Do you think it's a systemic problem, or is this just a nice to have?
I think it is a systemic problem. I think that it would be good maybe in every sort of entomology PhD to have like sort of like a dedicated part of the PhD that needs to be done that's like got some funding to spend, I don't know, 3 months at least being like, after you finish your collection, database the bees and have them in the museum. But that you also need to discuss with the museums because, as I said, they are running out of room, and getting more cabinets is expensive. So I guess it's having to, I guess, discuss with museums, can they even take on your collections, or universities. I know in the US many universities actually have collections. That's not a thing here in Australia. So I think it'd be really good to also, I guess, talk to universities and say, hey, like, you do have space. It'd be amazing to have an area dedicated to collections from your researchers, to store them, and really make any sort of entomology PhD. Again, there's not that many entomology PhDs either, or postdocs. Like, I've been struggling to find a postdoc, but any sort of university, say, really prioritize the value of collections, because I'm sure there are many people that probably collect and they just throw out their specimens, or they're sitting there in storage in a university somewhere. So yeah, I guess prioritizing collections and databasing, and putting aside some funding that's dedicated to making sure that these collections are preserved, because I really, I'm very passionate about bees and also like animal ethics, and I would hate to think that animals were sacrificed for no reason, like their deaths. Like,
No, I see that.
yeah.
Absolutely, and it's interesting. The other talks, of course, this is a natural aspect of collections, isn't it? Is that things in it are dead, and so making as much value as that as possible. If I could pass to David and Mieke, both of you talked about in your projects the challenges of joining data together, and how that was perhaps not unexpected for you, but to me in some ways a challenge. Can you talk about how much of a challenge that was, and how difficult it was to resolve? Was perhaps Mieke first, if you don't mind? Or...
Yeah, sure. Thanks, Martin. For us, I think it sort of comes down to the traditional sort of way, and that data has been kept, particularly in the museum, where it is very focused on specimens. But as sort of technology has evolved, it's allowed more for that linking of different records together, where back in the past it was like spreadsheet sort of style. You're very limited to individual sort of rows, like this is the thing, and you need to interrogate that particular row to understand it. But with more dynamic linking of data, what we can do now is really get the sense of the associations and understanding of those relationships more, especially with extra mapping capabilities as well. And I just really think that the technology is coming along, and we've just got to make sure our data is keeping up as well. So that's really where we'd wanted to bring that data out and make it more available to all those, and ALA is doing great in adjusting its data model as well to be doing more of that linking as well. And yeah, David?
Yeah, I think one of the key things is to plan ahead. So one of the things with many government grants and things is, and And lots of conservation projects in particular, they'll build collections, but they haven't necessarily planned for the long-term curation and care of those collections. And so that's something that really needs to be built into the funding model. And then I guess those collections, it's great if they can then come to a state institution where they have statutory protection. And I guess that's one of the dangers of unprotected collections like university collections, is that they don't have that long-term protection. So it's good if you are building collections to get in touch with the museums. And what we're doing is providing spreadsheets which give people the Darwin core fields to then match our database so that we can plan to accession that information and set it up so that their specimens are stored or housed or prepared in the way that they need to be for to smooth that transition into the collection. And the more work that you can do up front when you start building and plan for building the collection, the easier it will then be for them to be put into a state collection so that they can get that long-term protection and make migration onto things like the ALA easier. But yeah, taxonomy is massively underfunded and needs an amazing amount of support across so many different taxa, but especially in the invertebrate space. So yeah, no, that's an area that needs a huge amount of support. And in the vertebrate space, it's these tissue collections with the growing advent of genetics and things, and the expanded use of toxicology, and I guess drilling down on the different aspects of wildlife health and diversity. So to take eastern quolls for an example, they went extinct on the mainland. They were considered common in Tasmania, but we know that they've actually got a fair amount of genetic decline here, and as a consequence of habitat fragmentation and things. So even a species you might think is common, we need to have the genetic resources to be able to track those changes in diversity. And museum collections are quite unique in that we have collections built over time, which means we can look back at how genetic diversity may have changed. And of course, we're living in a slightly scary future now where people have started seriously discussing this idea of de-extinction. And of course, future genetic wizardry might allow such things, but it will only be possible if we have the collections built today when we can actually collect specimens for species so that we have that genetic diversity captured.
All good points. Well, sadly, we're out of time. So thank you, everyone, for your talks and for your comments and your thoughts. And thanks, everyone at home, for joining us. This talk will be available on YouTube afterwards, so feel free to watch it back anytime. And have a good day. Thank you very much.
Thank you. Bye.
Thank you, everyone.
Thanks.